Hi. Okay, let's talk about the immune system. And this is a topic that's become of great interest to a lot of people in the last year because of the COVID situation. So you'll recognize a lot of stuff in this chapter at like neutralizing antibodies and T cells and B cells and memory cells, antibody response, lots of phrases that we've been hearing related to coronavirus. So our immune system is designed to protect us from invaders, from pathogens, and our immune system responds to viruses, bacteria, also fungi, and even protists that are, or even parasitic worms and such things. And there are different parts of the immune system that respond to these. We have one part of the immune system that is a rapid, kind of a general response, and then we have a delayed response that is specific to each pathogen. So the rapid response is called innate immunity. The innate immunity can recognize general characteristics of bacteria and viruses and fungi and attack them. This even includes barriers like our skin and mucous membranes. Vertebrates have adaptive immunity as well. And adaptive here, the key there is the word adaptive, and it means that the immune system is going to be altered after an exposure to a particular pathogen and remember that pathogen for next time so that we can have a rapid response. And it's adaptive immunity that we're talking about when we're talking about vaccines, for example. So innate immunity is barriers, including things like mucus secretions from mucous membranes, which help trap and expel invading bacteria, fungal spores, and viruses. Also recognizing general proteins that are found in many kinds of bacteria and attacking them and reacting to them immediately without waiting for an antibody response. The adaptive immunity, this is antibodies. That's the humoral response. Antibodies are not cells. Antibodies are secreted proteins, and we'll talk more about those. And then also cell-mediated response, which is cells. And these are cytotoxic T cells and other cells that are responding specifically to a learned epitope or particular shape of a molecule on an invader that's been seen before or has been in a vaccine. This is the same video that was in the last chapter, circulation of white blood cells rolling along in a zebrafish, so you can watch that again if you want to. I'm not going to show it to you right now. So innate immunity can include barriers, but there are also phagocytotic cells that will consume bacteria, consume viral infected cells without needing the learned response of antibodies. There are antimicrobial peptides as well that can destroy bacteria. One of the cells, I think this is my favorite cell in the innate response, are the natural killer cells, which are also part of our cancer response. Interferons are proteins that are secreted that activate a response to invaders even without a specifically recognized invader. And this also leads to an inflammatory response. So if you have a wound, within hours you're going to have a noticeable inflammatory response to this. And it's not being caused by the adaptive immunity. It's being caused by the innate immunity, just responding to the wound and bacteria that are present in the area. So barrier defenses includes mucus. Mucus is secreted. You probably heard that term, mucous membrane. So all of the interior epithelial layers in our body, including your esophagus and your trachea and your pharynx and inside of your mouth and inside of your nose and inside of your sinuses, are protected not just by an epithelial layer, but by the secretion of mucus. Mucus is a protein mixed with water. The protein is mucin, and it, when mixed with water, forms a sticky substance that can just physically trap bacteria and fungal spores that end up in your respiratory system and in your digestive system. Goblet cells are the mucus secreting cells that are found in these areas, and you probably know that if you have if you get a viral infection or a sinus infection, these cells secrete even more mucus because they're trying to get rid of the invader. And that's why when you have a cold, you have a lot of mucus. It's part of the innate immune response. So some of the proteins that are commonly found in bacteria and viruses, and even the bacterial and viral DNA and RNA, are recognized by receptors on macrophages. And these are called toll-like receptors because they're like a receptor that's called the toll receptor. But don't worry about the name of them. But these are just general reactions. So if you have a wound in your skin, bacteria get in there, there's going to be inflammation just as a response to particular proteins that are present, or even just the DNA or RNA of the bacteria can bind to one of these receptors and trigger a cellular response, part of which is going to be inflammation, which is one of the purposes of inflammation is to attract cells like neutrophils to the area that are going to then engulf the invaders. Two main types of phagocytotic cells, and remember, phagocytosis is when cells engulf something in a vesicle that they're going to eat. Phagocytosis is cell eating. So we have a lot of cells, different kinds of cells that can do this. Neutrophils are one. Neutrophils are present in large numbers in your blood, and you may have noticed when you looked at the blood cell slides that you see a lot of red blood cells, and mixed in there you see these large cells that have a very noticeable nucleus. Sometimes, depending on the angle, you can see that the nucleus is kind of U-shaped. Those are the neutrophils, and they are looking for a signal to crawl out of the bloodstream and go to a site of inflammation. Macrophages migrate and different parts of the body. They're in the lymph, and they're also in your interstitial fluid, and they as well are looking for something to react to all the time. There are also dendritic cells. Dendritic cells are really important because these are part of generating adaptive immunity. Dendritic cells are called antigen-presenting cells as well. So you'll see antigen-presenting cells mentioned later. So dendritic cells, when they encounter an invader, take it in and then take some of the bits of that invader and present it on their surface. And then they go to a lymph node and look for T cells and B cells that can react to that particular invader. Eosinophils are another white blood cell, another part of the immune system, and these react to specific parasites. So eosinophils are important in reacting, especially to animal parasites and protist parasites. Innate cellular defenses involve natural killer cells. Natural killer cells are recognizing general characteristics of bacteria, virus-infected cells, and also cancer cells, and they destroy them. They release chemicals that kill them and then can consume the parts of the dead cells. So natural killer cells are a really important part of our natural cancer defense system. You might not know that the reason that young people get fewer cancers than older people, or one of the reasons, is because younger people have far more of these patrolling natural killer cells. They're looking for abnormalities. They're looking for cells that have mutations, that aren't presenting the right molecules on their surface because they've been mutated. Looking for cells that are presenting viral proteins on their surface, meaning that they've been infected with a virus, and then they destroy them. So we already talked a little bit about the lymphatic system in the last chapter. So the lymphatic system and the lymph nodes are an important part of the immune system. Lymph nodes are where many of these B cells and T cells are going to be sitting and waiting for a dendritic cell to show up with the right epitope on their surface to activate them, which is why very often if you have an infection of some kind, like if you have strep throat, the lymph nodes in your throat will be swollen and they can be painful as well. They're swollen and painful because there's a lot of cell replication happening within them, not because the lymph node itself is infected. It's swollen because it's activated. And it often means that that particular swollen lymph node is near an infection. There are also peptides and proteins that are synthesized as part of the innate immune response, and interferons. There's a whole bunch of interferons that have now been discovered. Interferons are proteins that are secreted by these immediate response cells. They bind to viruses, and they also activate nearby macrophages and increase the inflammatory response. There about 30 of these proteins have been identified so far, some of which bind to bacteria and lyse them, some of which bind to infected cells, that's cells that have been infected by viruses, and cause those cells to die as well, so they don't continue to make new virus particles. Because you remember, viruses have to use our own cells in order to replicate. So if virally infected cells can be recognized and killed, that helps protect from the viral infection continuing. So the inflammatory response, and this again, this is something we've been hearing about in regards to coronavirus, is because an incorrect or an improper inflammatory response is one of the things that can cause very severe symptoms with coronavirus. And in fact, when you start to feel symptoms from a viral infection, those symptoms are being caused by your body's own response to the virus. So it's not the viruses themselves that are, or viral replication, that's making you feel sick. It's your body reacting to those viruses and trying to get rid of those viruses. That's the symptoms that you feel. And one of the primary things, or one of the first things that's activated when there's any kind of an invasion, is histamine. And you probably have heard that term in relation to taking an antihistamine. So if you're, hey, go eat your food. Stop it. If you're having an allergic reaction, for example, which an allergic reaction, that is your immune system responding to inappropriately to something like pollen, which is not an invader. If you are allergic to pollen, your immune system is generating an inflammatory response to something that is not actually a threat to your body, namely pollen. Pollen is not a threat. Pollen spores or pollen that's in your nasal cavity is not a threat, and your body is reacting as if it is, though. And one of the things it produces is histamine. Histamine triggers that redness, swelling, dilates blood vessels, causes itching and pain. That's it's produced by mast cells and other cells. And that's what you're taking an antihistamine for. You're taking an antihistamine to reduce mast cells and macrophages and other cells that produce histamine. Get them to stop to lower that inflammation. So some of that is good. I mean, the histamine response, it has a purpose. One of the things it's doing is it's going to make the blood vessels, the capillaries around the area, leaky, and that's going to promote neutrophils and other phagocytotic cells that are in the bloodstream to ooze their way out and into the tissue. So very quickly, all of your immune system warrior cells are showing up at that site being attracted by the histamine that's being released and being helped to leave the blood vessel by the fact that this whole inflammatory response, including the histamine, is making the capillaries leaky. It's allowing fluid like lymph to flow out and into the surrounding tissue, and now you have swelling and redness and pain and itchiness. But that also means that the white blood cells are on the scene. So here we have a little wooden splinter penetrating through the skin layer. Of course, a splinter is going to have bacteria on it because everything has bacteria on it, unless it just came out of a package like a syringe at the doctor's office that they just pulled out of a sterile package. It's going to have bacteria on it. So skin is penetrated. Mast cells are going to release histamine. Macrophages that are in the area are going to react to detecting bacterial proteins and DNA and RNA and start to release cytokines and interleukins. Cytokines, there again, there's another word that we've been hearing lately with overreaction to coronaviruses causing a cytokine storm. So cytokines activate nearby cells, cause other immune cells to divide and be attracted to the area, and generally increase the inflammation. So all of this activity is going to attract neutrophils in the bloodstream to leave. It causes capillaries to swell and become leaky. That's the redness that you see. So we have an increase of blood flow to the area, which is exactly what you want to counter the invasion here. So we have bacteria that are now in the lower tissues of the skin, and capillary is now swelling up because of histamine and the cytokines. Neutrophils are now going to say, hey, something's happening, we're needed, and they're going to squeeze their way out into the tissue. And these are all, the macrophages and the neutrophils are phagocytotic cells that will respond to any bacteria and eat them. So wound healing will proceed, and the phagocytotic cells, neutrophils, will continue to clean up the debris, eat the dead cells, because some of the bacteria are going to be killed by interferons and other toxic peptides that these cells are going to secrete, and they will continue to clean up the mess. This is a little video of a neutrophil in culture crawling. So let's take a look at that.
Watch a neutrophil move toward a chemical attractant supplied in a glass micropipette. Neutrophils are white blood cells involved in defense against pathogens that respond to chemical signals via G protein-linked receptors. They are attracted to sites of injured tissue where pathogens may be found. Notice how the cell forms an actin-based protrusion in the direction of the chemoattractant. Credit: Henry Bourne, University of California, San Francisco Medical School.
And you'll notice that that crawling neutrophil looks very similar to an amoeba. And in fact, they're using the exact same mechanism. They are using the actin that's located in a big network underneath the plasma membrane to create pseudopodia, and then the cytoplasm follows into the pseudopodia. So that's the actin part of the cytoskeleton that's being used for that, exactly the same as in amoeba. So inflammation, hopefully it's just going to stay local, but it can also be systemic, and it can trigger fever. Fever is part of your body's defense mechanism against bacteria and viruses, and it has been shown that bacteria, especially the kind that cause like strep throat, are very, very sensitive to temperature. So an increase of a couple of degrees in temperature really does slow down the ability of these bacteria to divide. And if you've ever done bacterial cultures in a lab, you find out very quickly that, well, the bacteria that you're most likely growing in a lab, if you're using bacteria for any kind of purpose, is E. coli. E. coli are the workhorse of the molecular biology lab. They are very sensitive to temperature. If something goes wrong with your incubator and it's a half a degree too warm or a half a degree too cool, you come back the next day, your bacteria haven't multiplied at all. They're very, very sensitive to changes in temperature. So fever is a really good way of helping fight an infection, which is why a lot of doctors today say, you know, unless you have a specific reason to try to reduce your fever, as soon as you get sick and you get a fever, don't take medicine to reduce your fever immediately. See how you feel, because the fever is part of your innate response to an infection, and it's actually going to help your body to fight that infection. Now, if the fever is preventing you from sleeping, or if there's some risk of seizures, that's a totally different question, because sleep is also really important to help fight illness. So if the fever is making you completely miserable and you can't sleep, that's a different situation. But think of a fever as part of your body's defense, because it is. Sepsis and septic shock are life-threatening. You can have a completely systemic inflammatory response that is no longer helpful against attack and can in fact be a problem. Some pathogens can evade the innate immunity and even the adaptive immunity. Some can change the epitopes that they're displaying on their surface every 5 days or so that the adaptive immunity cannot respond, and confuse the innate immunity by changing what they look like. Some, like tuberculosis, can hide from the innate and adaptive immunity by encasing themselves in a fatty layer and walling themselves off from the body. And can remain in the body for years, kind of hiding from the immune system. So there are pathogens that can get around this system. So adaptive immunity is a specific response to something on the invader. And the second time you're exposed to that same invader, the reaction is going to be really fast. Initially, adaptive immunity, though, is slow. It takes about 2 weeks to ramp up a good adaptive immune response to a newly presented pathogen. And that's why vaccines are so helpful, because they give you that first exposure so that if you ever see that pathogen again, instead of taking 2 weeks to ramp up antibody production, now it's going to take like 48 hours to ramp up antibody production. So adaptive immunity involves receptors that can recognize the shape of specific molecules on the invaders. These specific molecules, or the shapes, are called epitopes. Epitope is just a fancy word for the shape of something, usually some kind of a protein. And the adaptive immunity involves 2 kinds of cells, T cells and B cells, and they're called that based on where they initially form. T cells originate in the thymus, and B cells originate in the bone marrow. And that's why they're called that. The thymus, which we saw during our dissections of the pig, it was much harder to see in the frog, but you probably found it in your pig, because in young mammals, the thymus is really big, and then it shrinks as you go through childhood, and by the time you're an adult, it's pretty small. So antigens are the thing that's being recognized by the antibody. Antigens are often proteins, but they can be other molecules as well. But they're very often, they are surface proteins. So every bacteria, every virus, their surface is peppered with these proteins. And for the viruses, they use those proteins to bind to our cells and invade the cell in order to reproduce themselves. For bacteria, they also have particular proteins on their surface that they are using for different reasons, to stick to each other, to stick to surfaces. T cells and B cells have receptors on their surface that are stuck in their plasma membrane that can recognize different antigens. So each B cell or T cell only recognizes one kind of antigen, and we have tens of thousands of different ones. So each one only recognizes one, but in a conglomeration, all of your B cells and T cells can recognize tens of thousands of different shapes. And it was wondered for a long time how this was accomplished, since especially once our DNA was sequenced, it was realized that we only have hundreds of genes that code for these receptors. How do we end up with tens of thousands of different antibodies? And the answer is because the DNA is rearranged as these cells mature. The genes are actually shuffled to make new genes. So each person has a unique assortment of these different possible genes for making these antibodies or antigen receptors when they're on the surface. The antibody is what we call it when it's secreted. So antigen recognition by B cells. So the B cells have a receptor that has heavy chains and light chains. And on the exam, I'm not going to ask you to identify these by name, but it's good to know these because especially if you're going on to take human physiology, you're going to delve into the details of this. And again, all of these physiology chapters that we're looking at, circulation, and we're going to do kidney next, I'm just kind of giving you an overview of each of these topics, and I know that if you're going on to take anatomy and physiology, you're going to go on to a lot more detail in these topics. So we're just trying to cover. So instead of focusing on the little details here, focus on the big picture. What do each of these types of cells do generally? How do they do it? Where are they found in the body? Where do they go? Instead of the little details of what all these different things are named. Okay, so here is one of those antigen receptors. So you can see it's kind of Y-shaped, and also antibodies, secreted antibodies are Y-shaped as well. And there are antigen binding sites, and these regions that make up the antigen binding site, these are the variable regions. These are the regions that are coded for by those genes that get randomly shuffled. I shouldn't say randomly, it's not really random, but that get reshuffled and remade as these B cells are maturing in the bone marrow during infancy. So that's how we end up with the ability to recognize tens of thousands of different antigens, even though we only have a small number of genes that code for these variable regions. So once the B cells recognize their antigen, eventually they're going to give rise to cells that are going to secrete antibodies. So an antibody is a secreted protein that's going to recognize that same antigen. And the abbreviation for antibodies, or immunoglobulins, because they're proteins, are Ig. And there are 6 or 7 different Igs. IgG is the major one. And also IgA would be the second most common one. So when we're talking about neutralizing antibodies against a coronavirus, we're talking about IgG. Those are 85% of your antibodies are IgG antibodies, and those are the ones that we're testing for. So if you go have a blood test to see if you've been exposed to coronavirus sometime in the past, they're testing your levels of IgG to test it for matching against a specific virus. And sometimes they call that an antibody titer test. So if you're going to go work in a hospital, you have to have antibody titer tests often for particular things like hepatitis B. And that's what they're testing for. They're looking at your antibody level for a specific IgG to see if you, in fact, are immune to, and they can tell very distant past exposures, like even childhood exposures. They can tell if you were exposed to measles or chickenpox as a kid using an antibody test. So here we have our B cell with its specific antigen receptor, when it recognizes a specific epitope, and this could be from an antigen-presenting cell, like a dendritic cell, coming to the lymph node where it is, once it recognizes that, oh, I match this, it's going to start to replicate. It's going to replicate, and then those daughter cells are going to produce antibodies to that specific antigen. Hey, come on. Enough. And there can be different cells producing slightly different antibodies against the same pathogen. So if you are exposed, say, to the new coronavirus, and you get sick, and they they look at your IgGs that you are producing against this virus, they might find an assortment, 4 or 5 different ones that actually bind to slightly different epitopes on the surface of the virus, and maybe only one of those antibodies is going to be what we call a neutralizing antibody, meaning that it's going to bind to the spike protein that virus uses to bind to cells and will prevent cells from being infected. So you're going to produce an assortment of antibodies to one specific pathogen, and hopefully at least one of those antibodies is going to be a neutralizing antibody, which will prevent a reinfection. Now, if you happen to not produce any neutralizing antibodies, maybe you have a very small exposure, you don't really get very sick, you get better, but none of the antibodies that your body produces is specifically a neutralizing antibody. That's how they believe some people are getting infected a second time. And then the second time they get infected, different B cells get activated, and now they produce neutralizing antibodies. So T cells as well do the same thing. They have proteins on their surface that recognize specific antigens. But the T cells are going to do something different. The T cells don't secrete antibodies. The T cells multiply and are part of the cell-mediated defense. But it's still adaptive because they're going to recognize the specific epitope. So here's a T cell, and you'll notice they also have a receptor that has a variable region that are generated in the same way by moving different parts of the DNA around in the cell to make different genes for these variable regions. So we can recognize with our T cells tens of thousands of different antigens, and they have a different reaction though. When a T cell recognizes its antigen, it's going to multiply. It'll be triggered to multiply just like a B cell. But those resulting T cells are now going to just directly attack the invader. That's why it's a cell-mediated defense. They don't secrete specific antibodies. They go seeking the specific invader and bind to it. And if it's a bacteria, they kill it. If it's a virus, they neutralize it. And they're also phagocytotic cells. So in the next video, we'll talk more specifically about the B cell and the T cell response to pathogens.